Rafe and I love hosting these events is because the wide variety of science that we see presented here. Everything from the origins of the cosmos to the origins of life on Earth to RNA. And tonight we'll be hearing about sea mammals, those creatures that we share the ocean's edge with. Speaking tonight will be Carolyn Casey, who is a recent Ph.D. from Santa Cruz. Congratulations on that, Carolyn. And she will be talking about communications between sea lions. And then after her will come Dan Costa, who's been recently appointed as the head of the Maritime Institute at Santa Cruz and has been working in this area for over 30 years. We'll have a question period after Dan's talk, and for those of you who are watching online, you can send in your questions through the comment. So Carolyn, please join us.
Hello, everybody. Show of hands, how many people are UC Santa Cruz graduates here? Wow, okay, excellent. I would like to say that I did my undergraduate, my master's, and my PhD all at Santa Cruz over the past 10 to 12 years or so. So I'm really excited to be here. Thank you for organizing the event and for inviting me here. I'm going to be sharing some of my dissertation research and also just talking generally about northern elephant seals. And sharing with you what I think are some really interesting aspects of their behavior. My talk is called Combining Technology and Ethology to Explore the Secret Lives of Seals. And I was a recent graduate student with Dan Costa, and I now work for a new nonprofit called the California Ocean Alliance over in Santa Cruz. So broadly, I'm really interested in the way that animals interact with one another, how they solve problems, and how they kind of navigate their social dynamics with one another, either to find food, or to rear their young, or to compete for mates. And acoustic communication is a really fundamental aspect of interactions between animals. So it basically is a part of nearly all life history events, from parent-offspring interactions to male-male competition. And over the long history of animal communication, there's a lot of people who happen to be interested in this topic. Birds have served as the primary model for studies of the evolution of vocal signaling in animals. So field experiments have been paired with very careful observations in captivity to answer questions about evolution, ontogeny, behavior, and the functionality of what these calls really mean. So most of you probably have heard bird song and know that it's really important for male-male competition and territoriality. And unfortunately, for a lot of the rest of the animal kingdom, that type of detailed information is lacking. So up until now, bird song have served as sort of the model system. And so I've been really interested in marine mammals over the course of my graduate and undergraduate career. I've worked at the Long Marine Lab for a long time, and marine mammals are a group of animals that make their living in the sea, and some individuals are tied to land, and others, like cetaceans, like dolphins and whales, have to spend their entire lives at sea. But they are incredibly dependent on acoustic communication underwater. So because the visual cues and light attenuates quickly at depth, the underwater characteristics of the sound or of the water environment allow individuals to stay in contact over large distances using sound. Particularly low frequency sound can travel really far. And to sort of orient our minds to an acoustics talk, I've prepared a little montage of marine mammal sounds that I'm going to play for you. If we could turn the sound down slightly for this, there's one call in particular that is a little bit disturbing. And I'll try and point out what these animals are as they come up. So this is an elephant seal, female in captivity. A polar bear. This is the one that hurts. This is a baby sea otter trying to contact its mom. This, anybody know what this is? It's a walrus. It's the underwater display of a walrus. Yeah, elephant seal. This is a male elephant seal that I recorded. This is a whistling behavior by a walrus. So they have these really mobile lips, and they can actually form whistles. This is a trained animal in captivity. Anybody know what this is? What did you say, Dan? A Weddell seal. Yeah, so the Weddell seal producing an underwater display. It's a leopard seal. That was also a walrus. So walruses are really freaky. They make a lot of amazing noises. That's a harbor seal. Actually, a harbor seal that lives at Long Marine Lab. This is a baby elephant seal. And then the last one, which happens to be my favorite, is a combination of Weddell and leopard seals together in the Antarctic, which is really haunting. They make some really beautiful sounds. So you can see that there's this huge diversity of sounds that are produced by marine mammals in particular. Let's see if I can advance here. Oh, I don't want to start that again. And I would say about half of them were in air and half of them underwater. The animals that rely on underwater sounds, whales and dolphins, a lot of seals produce sounds underwater because they mate aquatically. Sea otters produce sounds in air, as do polar bears, obviously, and then sea lions do a lot of their behaving, as many of you probably know, in air as well, so on land. So it kind of varies. The study of marine mammal acoustics was really a result of underwater listening technology that was developed by the Navy during the Cold War. And so they were looking for, Russian submarines, and it's a little bit difficult to see. Oops, there we go. But there's these little red stations, hydrophone stations, that they put all over, extending through the Atlantic and the Pacific Oceans to basically listen for military operations off the coast. And they had to be able to differentiate between what were marine mammal sounds and what were potential Navy sounds. They detected what was called the Jezebel monster. They had no idea what that was, but that's what they recorded it as. And later it was actually found out to be a fin whale. So this was kind of where the marine mammal acoustics field was sort of born, was out of this necessity to be able to differentiate between these two. And so if you are an acoustician like myself, or somebody who's interested in animal behavior, you can imagine going out in a boat and pulling, a hydrophone behind your boat and listening for the underwater sounds of, say, a humpback whale who's producing calls. And it's really great. I mean, we love humpback whale song, but unfortunately, because the behavior is occurring below the surface of the water, we don't know what the behavioral context is of that signal. So he's emitting the signal, but who are the other individuals in the environment that they're calling to? Is this a male or a female humpback whale? And how old is it? I mean, these are the factors or the questions that really allow us to get at that detailed information that we've been able to gather from things like birdsong. So the field of marine mammal acoustics has been basically describing the types of sounds that marine mammals make, but not really actually answering questions regarding function and these deeper questions that I'm really interested in that we've been able to do in birds. And so hopefully by the end of this talk, I will have convinced you that northern elephant seals are an incredible model for the study of sound, and in particular, looking at the function of specialized acoustic displays. And what I'm going to be talking about are male elephant seals because they're awesome, and they produce some really amazing sounds in air. And elephant seals are unique among seals in that they breed on land, so they're one of the few species that haul out to breed instead of breeding aquatically. And because they haul out in predictable locations each year. How many of you have been to Ano Nuevo State Park? Great. So you've seen them, hopefully in action. It allows us to really keep track of focal individuals, record their sounds really well, and see the behavioral context in which these sounds are being emitted to other individuals on the colony. So I think northern elephant seals can serve as an incredible model for studies of animal communication. And that's what I've dedicated the past 10 years of my life studying. My PhD didn't take 10 years. I was, just to be clear. The fact that we even have northern elephant seals today is incredible. Some of you might be familiar with their story that they were hunted to the brink of extinction in the late 1800s because their blubber was rendered for oil. And so they were essentially decimated along the California coast in Mexico where they were previously breeding. And then the last remnant population was found on Isla de Guadalupe, which is about 150 miles offshore from Baja. And there are thought to be, the number is debated, but between less than 50 individuals left in the entire population. And so miraculously, they survived and evaded hunters, and were able to repopulate their historic range. And so now we have elephant seals recolonized San Nicolas Island in 1938, then went up to San Miguel Island in 1950, and then in 1961 they recolonized Año Nuevo, and then have since then been even extending further up north, and now there's a new elephant seal colony at the Lost Coast that has just been established within the past 3 or 4 years or so. So they're continuing to expand. And so most of you probably associate male elephant seals with these very brutal fights that take place. And so it's true, during the breeding season, males do arrive first, and usually between the end of November, early December, and they fight to establish dominance hierarchies, and these hierarchies determine access to these big groups of females. So one male is responsible for mating and guarding, between 50 to say 75 females at one time. So that's a very lucky male, but not everybody gets to be a dominant individual, and so they really have to duke it out in the beginning. But once they've established this dominance hierarchy, fights like this are really rare. So it really only happens in the beginning of the season. I'm just going to show you the costs that can be associated with fighting and chasing. So this is an alpha male, one of those males who gets to control everything, attacking a unsuspecting subordinate male who's gotten too close to his females. And so this was taken with a slow motion red camera from National Geographic, so you get the blubber action, and you can also get how intense these interactions can be during the breeding season. They have extremely big canines. So the more dominant individuals are typically running around doing this for the majority of the breeding season, which extends, some of them are there until March. So from December until March, they're fasting. They're defending their harem, they're mating with females, and they're running around like loons trying to chase out all these other males that didn't get a partner for the breeding season. So the females arrive after the males. Females arrive pregnant, like this picture on the left, and then within a few days of arrival, they give birth to a single pup that they take care of, between 25 and 30 days or so. So the females kind of group together, and then the males guard these groups of females. And so this is kind of what a harem looks like. For those of you who have been to an elephant seal breeding colony, it kind of looks like a cluster, but then when you look closely, you have one guy who's in the center who's in charge of everything and keeping eyes on everybody all the time. And then you've got some beta, what we call beta males that are on the outskirts that are sometimes tolerated by the alpha. Maybe you could be there for a little bit, but don't get too close, and definitely don't mate with anybody. And then you have these kind of subadult peripheral males who are younger individuals that are on the colony and they're lurking around, but they're completely excluded, usually by the betas. So it's this really interesting system of males that are interacting with one another frequently during the breeding season. And this is a shot that was taken at Piedras Blancas for those of you who have been down there. And this would be considered his harem. And so because of this intense intersexual competition between males, it's led to the evolution of extreme sexual dimorphism, where males are significantly larger than females. They have this long fleshy nose, the proboscis, and this really callous chest shield. And females are much smaller, and usually you have to be very big in order to be very dominant in this system. So even though there's a lot of fights that occur between competing males, most of the exchanges are resolved through the use of these ritualized displays that include vocalizing, slamming their chest on the substrate, and then also some visual posturing. So when I first started working at Año Nuevo, I became very interested in these acoustic signals that were being emitted during the breeding season, and what are they saying to each other? That was basically my Ph.D., just trying to figure out what is it that they are conveying to one another during these ritualized exchanges. And to show you how effective these calls are, the animal on the left is calling out, and you could turn it up a little bit now, we have no more intense signals. And this guy's all the way over here, and yet he's moving. And so this is a great way to conserve energy for those alphas. They don't have to run around and chase and fight, which is really good. And so how many of you are familiar with Bernie LeBoeuf's work? Some of us. Okay, great. So I started my graduate career, and I read some of his papers. You know, he was one of the first people to really explore the social lives of elephant seals and males in particular. And, you know, he was a brazen guy who was working in the field. And I read this really interesting paper called Dialects in Elephant Seals. And so he evaluated the vocal behavior of male elephant seals at these colonies right after they had been recolonized. So in 1969, this paper was published. And he looked and recorded males at each of these sites. And what he found is that within a site, if you were a male from Año Nuevo, for example, so the top three, what we're looking at are basically spectrograms. So on the x-axis, you have time, and on the y-axis, you have frequency. And if you were from a particular island, then you sounded like all the other males on the island. Everybody had a dialect just like British versus American dialects. And then if you went to Isla de Guadalupe, you sounded much different. And so this was actually the first example of dialects being found in marine mammal, or I'm sorry, in any animal species other than humans, which was really cool. And nobody had touched this work since 1969. And so I thought this was really peculiar. You know, the population has really exploded. And so I was really interested in retracing Bernie's footsteps and trying to establish whether or not these dialects have persisted as the population has recovered from near extinction. So my first step was to go out and actually record the calls of modern day males and compare them to Bernie's actual data. And then we wanted to test the prediction of whether these original dialects basically were maintained over time, or if they just were kind of a byproduct of the way they recolonized their original sites and have since disappeared. And then we also wanted to just evaluate what are the factors that actually lead or contribute to geographic variation in acoustic calls between these different locations. So I went out and I actually revisited all the places that Bernie had sampled. And just to give you an idea of what the populations have done at these locations over time, you know, basically most of them have grown, some really quickly, like at San Miguel and San Nicolas, and Año Nuevo has increased and then kind of tapered out. And so you can see when the historical recordings were made and when I was going to be making my recordings. And at each site, I recorded the calls of male elephant seals. And this is a really ritualized display, meaning that they sit up and they call, and they usually don't stop once they start calling, so you can move in safely, record them, and then walk away, although I wouldn't recommend doing this, but you can do it. And then as soon as we got to a colony, we would record their calls, but we would also mark them to make sure that we weren't recording the same male twice. And so to do that, we use black hair dye that you use to dye your own hair. And we mark them on their backs while they're resting or sleeping. And they're kind of like a VW bus in that they can't see like directly. They have a blind spot, basically. So you can kind of sneak up on them when they're sleeping. Okay, so then Bernie, I had never seen a reel-to-reel player. I'm sorry, I hadn't. And so he brought all of his reel-to-reel tapes to me, and he was like, here you go. Here are all the historical recordings that I made in the 1960s. You can go through and verify my results if you want and reanalyze them using a similar approach that you would use. And I was like, great. So I had to convert all of these reel-to-reel, it took a long time, recordings, and then I compared them to the contemporary recordings that I had made at these same sites nearly 50 years later. And then we measured a bunch of parameters, but it was about 315 hours of historical data that I went through. And we measured a bunch of the temporal features of each of the calls. And what we found was really interesting, and I don't know if you can see this, but these are 3 calls from Año Nuevo from the 1969 study. So I basically reconfirmed his original results. They sound identical, and you can kind of see visually that these calls are very similar to one another, yet these are 3 different adult males. And the contemporary males now have incredible variation in their displays. And so each male sounds really distinctive from one another. You have like doublets or triplets, and I'll get to playing these calls in a second. I just don't want to run out of time. But it was this incredible explosion of diversity within a population over a really short period of time. We're talking, maybe 6 generations of elephant seals, where you went from everybody sounding identical to everybody sounding really different from each other. And this is just, of course, of the males. And so after 50 years, the dialects that Bernie had originally reported had completely disappeared. And modern males now exhibited an increase in between individual variation, just meaning males were much different from one another. And it's really difficult to say why. Everybody's like, well, why did this happen? And it likely has a number of reasons, some of it being patterns of dispersal, but other reasons have to do with the next phase of this talk, which really gets to what is the function of these displays? Why might they sound really different from one another? And really what I want to stress here is that the well-documented history of the northern elephant seal really started at UC Santa Cruz, led by Bernie, continued by Dan, and now I'm trying to pick up where they left off, builds off of this data set and has allowed us to ask these really interesting questions about the evolution of acoustic communication in this species. And so if we go back to this video that I showed you, now imagine that this male who's vocalizing has a call, and it just sounds like a drum beat. But know that he sounds very different from his neighbor. So you might ask yourself, well, what could he possibly be saying? Does anybody have any ideas? Like, get away, yeah, get out of here, right? But know also that these males' calls, they don't vary at all. They're very stereotyped. So the male will call when he's really angry and sound the same, or just kind of angry, sound the same. If he's vocalizing at somebody who's very threatening, he sounds the same or less. So it's incredibly stable. But there's something within the content of that signal that's telling this guy to run away. And males are really different from each other. So we really wanted to understand what kind of information was encoded within the adult calls of male elephant seals, and also ask the question whether males can perceive differences between individual acoustic displays. Like, can you tell the difference between Sam's call and John's call, and do you respond differently depending on who the individual is? So to do this, we mainly worked at Año Nuevo, but we did some additional work at Piedras Blancas, which is located about 300 miles south of Año Nuevo. And again, we marked all the individuals within our study. We wanted to really get to know who these males were and how they related to one another within the breeding season with respect to dominance interactions and also proximity. So this is us, drawing on them again and putting these nice clean marks so we can safely observe them at a distance. And what we did is we watched all the social interactions between competing males. And for every time we saw males interact, we noted who the winner was and who the loser was, how far they had moved, and whether or not they had vocalized during that interaction and the intensity of that interaction. Like, did they fight or did they just sit up and vocalize and run away? And then at the end of the season, what we had was a list, thousands of interactions that we had observed, and the outcome of those interactions were fed into this ELO rating approach, which is actually used for 2-player games like chess. It was actually used by Facebook in its early years of the company, but it essentially detects the probability of one individual beating another based on their relative dominant status to one another. And so you can give a quantitative score to each male within our study, so you can get away from the subjective classification of alpha, beta, peripheral, and give every male a number on the colony that indicates how dominant they are. So 2,000 was the highest score, and 9R, who was a lowly, peripheral male, had only, about 1,000 or so. So a nice way of comparing individual dominance ranks. And of course, we obtained acoustic recordings of all the males at this site. And we also measured all of our males. So we took these photographs that were calibrated with this meter-long measuring stick. And when they were resting, we were able to obtain these photographs and then measure their perimeter and also their height and their length, and tried to get an estimate of mass based on these measurements. And so what we found was that, consistent with our earlier findings, males really sounded different from one another at the Año Nuevo colony. So you can have a male like 3C, adult dominant animal at the Año Nuevo colony. Very staccato, regular call. And then you could have a call like from my favorite, X579. beautiful. So some of the features were correlated to the size of the animal. So we measured a lot of the acoustic features within this call. And so what we found out was that really there wasn't like a particular call that conveys the size of the animal or the dominance. So it's not like the staccato call is the dominant male call. And or that X579's call has any correlation to how big he is. So there's no honest information contained within the calls, which is interesting because for other animals, the bigger you are, the lower your call is going to be, and you can actually perceive size, and that could be used as interesting information. With these guys, there was no kind of pattern of that that became obvious to us, but in order to really answer that question, we had to do a set of experiments to test that. So we wanted to really test whether males could perceive differences between individual males and respond differentially. So we saw in the field that males responded differentially to their known competitors. Sometimes they attacked and sometimes they retreated, but we needed a way to experimentally address this and figure out whether there was something contained within the signal that indicated size or dominant status, or alternatively, if these calls served as individual signatures that males were learning throughout the breeding season and remembering. So each male maybe has his own name. So that was kind of our running hypotheses. And to do this, we had an experimental design where we would record the calls of males in the field and then turn them into basically soundtracks or waveforms that we would play through a speaker and place about 7 meters away from the target animal that we were testing. And then we would sit on a dune and measure the behavioral response of that animal to the sound itself. So the speaker would emit a call of a familiar competitor, and then we would just measure whether or not they responded differentially depending on whether the competitor was more dominant or less dominant to the focal animal. So I'll go through an example of that. So if you take an animal like 16R, who is this mid-ranking beta, and when you played him the call of his most dominant rival 2S, we would expect that he would retreat from the speaker because that's what we had observed multiple times in the field. Whenever he heard his call, he ran away. Conversely, if he heard his subordinate rival who was most familiar to him, he should approach and attack the speaker, consistent with what we had seen. So we did this with 10 males, and each of them received these 2 treatments. And here's an example of 16R receiving his most dominant rival. This is an older video, so the quality's kind of bad. The speaker's in here. He's there. Now the sound is beginning. This is his most familiar dominant rival. He hears it. He's assessing. And mind you, there are no other males within the vicinity. This is just the response to the playback. And this is a huge animal, likely like 32 to 3,800 pounds running. So clearly that is a very salient signal that's conveying something very scary to him. But we had to make sure that 16R wasn't just a wimp and ran away from all the calls that we played him. So we did give him his most familiar subordinate rival. And I'm just going to fast forward this video a little bit because it takes a while to get going. He actually doesn't respond immediately to the playback speaker. So the playback speaker is up in this right-hand corner here. And then the target animal is here in the left-hand side corner. He's actually attending to the signal. He's listening to it. It's a big buildup. And then he does decide to respond aggressively to that signal. So these results were consistent across all of the animals that we tested. When they heard their most familiar subordinate rival, they would approach and conversely run away when they heard their familiar dominant rival. But we needed to make sure, so that does answer the question, yes, they can differentiate, but doesn't really get at, is that based on familiarity, or is there something in the call that's conveying something scary or less scary? So to get at this question, we went down to a new breeding site, Piedras Blancas, and we tested 10 males that had never had experience with those individuals at Año Nuevo. And so if there's something encoded in the signal, like size or dominant status, then they should respond similarly to the calls of the males up at Año Nuevo. However, if it's really based on individual experience, like if I say to you, Sam, but you don't know who Sam is and have no association with that, you should be like, I don't know what to do. I'm just going to sit here. So we did that same experiment with 10 new males at the Piedras Blancas location, and they didn't respond at all. So they wouldn't respond differentially. They just kind of sat up, stared at the speaker, and laid back down. So this means that males are learning and remembering the calls of up to 40 individuals within their social network and deciding to attack or retreat based on the outcome of previous interactions with those individuals, which is a huge mental feat for a male elephant seal, and I think that they're pretty smart. So just in summary here, adult males produce individually unique acoustic displays during the breeding season. Hopefully next time you go out to Año Nuevo, you can listen to those calls and see if you can tell the difference between adults. They function to control the movement of familiar rivals with minimal cost of conflict, so it's really effective in cutting down on aggressive fighting between individuals. And they don't appear to convey resource holding potential like size or dominant status to the receiver. And it appears that they really remember the identity of their rivals based on these interactions that they're having with one another over the breeding season. And I'm just going to throw these up here. Basically, in summary, the calls have really changed over the past 50 years. I think the reason that they have changed is because there's been increased competition at these sites, and it pays to be different. So with more animals at one site, it's harder to keep track of everyone, and so acoustically, they're becoming more dissimilar from one another, so they can figure out and keep track of who's who. This is my running hypothesis. And really what I want to stress is that this work has only been possible based on these long-term studies of elephant seals that have been conducted through UC Santa Cruz at this site at Año Nuevo. And I'll just end with a plug for a camp that I teach with my husband, Ari Friedlander. It's called the Marine Mammal Scientist and Training Program. It's taught to high school students during this summer. We're offering 3 courses where we're taking students into the field and giving them the skills that we received as undergraduates, but maybe not as high school students. There's scholarships available, so if you know anybody who'd be interested in this type of program, you can come talk to me after this talk or visit our website, californiaoceanalliance.org. And I think that's all for me. So thank you, guys.
I wasn't sure if it would be good to follow or precede Carolyn. She gives me too much credit. Yes, she was my graduate student, but she worked predominantly with Colleen Reichmuth. I gave her the elephant seal background, and Colleen was her bioacoustics expert, which I certainly don't have that level of skill. But that also speaks to the advantage of being at some place like UC Santa Cruz, where we have a variety of different investigators, and we work together in a way that the sum of us working together is much more than any of us could accomplish on our own. So I'm going to talk to you about, Carolyn talked about, what I really think about is the work she did is sort of a renaissance. Murray LeBoeuf started elephant seal work in the late 60s, early 70s. I came along and we started looking, we did work on the beach, but then we started going out to sea and studying what the animals have been doing at sea, and that's what I'm going to talk about today. And Carolyn told you about some of the really exciting work that we're now doing, that she and others are doing on the beach, and I'm still out there in the ocean worrying about what these guys do. But the other part of the story today is to talk about special places. And there's 3 special places I'm going to talk about that really show the importance of having a research site that has something unique about it that allows us, in this case, I'm going to talk about the development of new technology that allows us to understand what these animals are doing. And that first place is the Antarctic, and that's where my postdoctoral advisor, Dr. Jerry Kooiman, I went there in 1978, 1979, and this is a Weddell seal. And so the first part of my talk, I'm going to take you quite short trip to the Antarctic, and then take you to a very special place that Carolyn just talked about, which is Año Nuevo, and then I'm going to finish with another really exciting place, the Galapagos. So I'm going to talk about, this is the outline where we started in the Antarctic, taking us to Año Nuevo. What have we learned? Studies of where the animals go, what we've learned about how they do what they do, look at how we study their foraging behavior, how we're now using animals to study the ocean, and then I'll take you to studies of other species. So let's start with the Antarctic. This is a particularly amazing, special place. Go back to the 1960s where Jerry Kooiman went there, and nobody knew what these animals did. They actually shot them for food for the husky dogs that they use to traverse around. But what was amazing about these animals is they live here throughout the year, and they're very accustomed. This is a natural hole in the ice that this seal is occupying. So these guys are used to going and diving from little holes in the ice and then going out and swimming around. And Jerry Koyman took advantage of that, and he put a hut over. How does this work? Is it? It's not working. Top one. That's why it wasn't working. So there's a Waddell seal there, and he's got a hut. And this was the amazing technology of the day. It was a kitchen timer with a glass, a round piece of glass that was smoked with soot, and then it had a Bourdon tube that is a pressure sensor that would scribe, as the kitchen timer went for an hour, it would scribe a piece of the diving pattern. So taking this situation where we have a seal that naturally likes holes, putting a hut over it, and then the seal is moving from this hut because he's now taken the seal 10 miles away from where there's all these holes in the ice, and started to deploy these really advanced instruments like that kitchen timer. And so that then went to the next stage in technology when I actually was fortunate enough to join his lab, when we used this amazing time depth recorder, which used film from spy satellites, and we had there's a light emitting diode at the end of this arm, and as the pressure would go down, this arm would go across and make a record of the dive. And so we'd get this record that looks like this. It's that light emitting diode. We would amplify this, make a printout of it, and then we'd make these rolls of paper that we'd run down the hall, and it would take us 24 hours to hand digitize these records. That was the state of the art in the late 70s, well, in this case, it was 1983 was the first one we put on an elephant seal. So that was, with first time depth recorder, and this was a picture of that device on an Antarctic fur seal, and this is the kind of thing we're looking at today. This is a Cape fur seal where everything is done electronically, and Carolyn talked about using those tapes that she had to use a reel-to-reel, and I'm showing you the reel-to-reel age of time depth recorders. But this technology is rapidly developed, and so now I'd like to take you to Año Nuevo and show you this amazing place. And so Año Nuevo is almost, not quite, it's a little bit due southwest of us. And what makes this an amazing place is it's a 30-minute drive from the campus. And so Bernie LeBoeuf's been working there, started there in the late 60s, early 70s. I've been working there on and off since I was a graduate student in the late 70s. And it's allowed us to, in this picture, are undergraduates and graduate students, and we have a long-term data series. Now, like in the Antarctic, where he had a captive natural situation where the animal had to come back to an ice hole, but that was part of its normal life, we have elephant seals that sit on the beach. We can put these instruments on them, and you can see the instrument right there, and they'll go to sea and come back. And so that enabled us to try things and new equipment, new technologies that nobody had ever tried before. And so that's what we've been doing. The first thing we did is, if you would have read, this was an up-and-coming book when I was teaching marine mammal biology. If you looked at this textbook on pinniped biology, that was the distribution of northern elephant seals. That's because that's where we could look for them. This is where we could fly a plane or drive a boat, and so we said, here's the range of northern elephant seals. When we put these tracking devices on them, we found out that their range is pretty much the North Pacific Ocean. And then more recently, when we've had the technologies a little bit more accessible, a little more cost-effective, we've been able to put these on. We now have over 600 tracks of different females that we've been looking at for the last 15 years. So we have this long-term data that we can now ask much more interesting questions about what these animals are doing. This is an animation that shows you when the white worm is long, the animals are moving fast. When it's almost a dot, the animals are either not moving or are staying in one spot. So what we realized from this is that the animals are feeding out here, and they're migrating back to Año Nuevo, or in this case to the San Benitos Guadalupe Island, which Carolyn talked about as well. And they have 2 trips a year, the post-breeding trip, which lasts 3 months, and the post-molting trip, which lasts about 8 months. So each one of these trips is is these are multiple years now of elephant seals that are migrating out into the North Pacific. So this is what the dive record looks like if you take each, this is nighttime, daytime, nighttime, daytime. So this is 4 days in the life of an elephant seal. This is depth. And the first thing you can notice that these animals, there's no period of time where they're not diving. So they're constantly diving. During the daytime, they're diving deeper to 620 meters. In the nighttime, they're much shallower. And they just dive continuous for that entire 8-month period of time. They spend almost no time at the surface. In fact, they spend about 20 to 25 minutes underwater and 2 to 3 minutes at the surface. So 80% of their time is spent underwater. Now, one of the things that one of my current students is doing, people ask, well, when do they sleep? Well, we don't really know. And so she's got access to state-of-the-art technology where we can put a little EEG sleep measuring device that we can attach to the head. And she hopes to be able to, just like in that picture up here where we have satellite tracking devices that will be able to measure whether or not these guys are sleeping, or probably more like when they do sleep at sea. So some of the amazing thing about these animals is here is a sequence of dives, an hour dive, 50-minute dive, an hour and 43-minute dive, an hour and 10-minute dive. And what's amazing, in each one of those dives is less than 2 minutes. The time spent at the surface is less than 2 minutes. I can go into a lot of discussion about our understanding of the physiology of diving, and all I can tell you is this animal didn't read any of our papers. So we really are trying to figure out exactly what is going on here, and there's actually a group back in Santa Cruz, again, a team of investigators from a number of different institutions who are developing physiological tags that we can put on these animals and start to get a better understanding of what's happening. And here's an example of such a tag. This is the dive record of an individual that we instrumented with some very sophisticated equipment, took it down to Monterey Bay, released it, and it swam back to Año Nuevo and recovered it. And so this is the diving pattern. This animal's going about 150 meters. And then we have indwelling catheters that are measuring the blood oxygen levels of that animal during the dive. And so you can see that the animal starts the dive initially. As the animal's diving, the oxygen content of the blood gets higher because the concentration of the air, the oxygen in the lungs is increasing. And so initially, the oxygen content of the blood goes up. But then during the dive, it declines, and it gets to levels that this line here is where we would black out. So this is where you or I, if we held our breath this long, we'd pass out. This elephant seal is functioning well below that level. At the end of the dive, it comes up, and you can see that it reperfuses, and when the lungs function, it starts to recover. This figure shows a picture of how that works, such that in three-dimensional space, the animal comes up from across Monterey Bay, and you can see the dives. It's got oxygen-rich. The dive proceeds, the oxygen's depleting, and then it comes to the surface, and the oxygen is almost out, and then it hits the surface and starts breathing again. So we have technology that's now using medical instrumentation that we're applying to these animals as they freely move out in the ocean to figure out how they work and what they're doing. Now, I always say it isn't rocket science. Well, I love this because Jessica Mayer was a Ph.D. student from Scripps, but she came up and did half of her dissertation. The work I just showed you was out of her dissertation. And she finished her work with us and Paul Panganis, then went on, did a postdoc, and she's now an astronaut. So it may not be rocket science, but for some people it leads to rocket science. So some of the other things we've been able to do, because we have this large data set, we do get a few females that don't reproduce, or something happens to them. We put the tags out, and this is looking at the dive duration. So this is how long the animal is diving. And this is just showing the annual cycle of an elephant seal. This shows the red is females that don't have a pup, and this blue line is females that do have a pup. And so what you can see here is that when the female starts, there's no difference. And as the fetus starts to grow, the female with a fetus has a lower dive duration. It can't spend as much time underwater. And that probably makes sense because it's got another little body inside of it that is sucking up some of that oxygen. So this is just by looking at behavior. And because we had this large data set, we had females that we knew that came up on the beach without having a pup. And we were able to compare what they did at sea to females that did have a pup. And one of the beauties of doing work like this is that we can do studies with elephant seals that can give us insights to other species where you can't do all the manipulations that we're able to do with elephant seals. So we can learn a lot from these animals to then ask questions about species that are harder to work with.
Individuals in that graph?
How many what?
How many individuals?
We have at least, well, for the females that have pregnancy, we have literally hundreds. For the females that weren't pregnant, about 10 or 15, at least 10 or 15 of those. So one of the other things is early on, one of our colleagues from Japan who we'd been working with said, hey, what's these funny dives? The animals go down to depth. Actually, I skipped early on, we would see these records where we would think that these looked like good feeding dives, where the animals are going up and down, and we postulated that those were periods where the animals were foraging on fish. But then we'd see these other dives where the animal would go down to depth and then stop and sink, and then come back up and breathe, and then go down to depth, stop swimming, and then sink. And so we started to think, well, these must be processing dives, because these animals never spend any time at the surface. When you have a big turkey dinner or a big Thanksgiving dinner, what do we all want to do? Go sit down and sleep. Because all the blood's flowing to your GI tract. So we think that's what's happening here, is that after a long series of feeding bouts, the animals are, instead of spending time at the surface, because it might be a dangerous place to be, a killer whale or a white shark might come and get you, spend your time at depth, but spend that time putting the oxygen to your GI tract instead of to your muscles. And so we think that's what's happening here. Now the cool thing about that is that because the animals are sinking, and we verified this with accelerometers, they're sinking directly as a function of their body size or their fat content. So skinny animals are going to sink rapidly, and fat animals are going to actually, in some cases, will float up. Now what's cool about this is it means that while we're studying the animals at sea, we can figure out where they're doing well, where they're getting more food, where they're successfully foraging. And so we can take those data now and look at where the animals are feeding. Here's the tracks of the animals at sea, and here's where they're doing lots of drift dives. And here's where the animals, remember I said when they're transiting that they're not moving somewhere? So the areas where they're spending a lot of time in one area is also the places out there where they're doing a lot of these drift dives. And so we're beginning, this gave us pretty good confidence that this is where the food is, this is where the animals are doing quite well. Now again, our Japanese colleagues come into this story a lot because they always come up with the neatest and greatest little devices. And this here is a jaw accelerometer that measures when the animal opens and closes its mouth. And these red dots are periods during the dive where the animal has actually gone after fish. And we calibrated this by looking in the lab in animals in captivity, and we calibrated what that accelerometry event would look like. And so you can see that sure enough, here's that processing diving area. The animals aren't opening and closing their mouth. The bottom of the dives, here's another, you can see, here's the areas where the animals are actually going after food. So now we can monitor where and when these animals are actively foraging. Now one of the cool aspects of this is if we look at, again, you have lots of records, we find some of the larger females are making unusually deep dives. So most of the diving is in this 400 to 600 meter range, but we get some individuals that are diving to those kinds of much deeper depths. And it looks like those animals, they're not having anywhere near as many jaw opening and closings, so we think they're feeding on larger fish. But what's even more interesting is I'm about to tell you about how we now have little cameras that we put on the seal's head that are triggered by that jaw accelerometer. So when the animal starts opening and closing its mouth, we take pictures. But what's interesting is this fish that we took a picture of isn't swimming away. One of the things that occurs in that deep part of the ocean is it's the oxygen minimum zone. And this oxygen minimum zone is an area where the oxygen has been depleted, and it's known that some fish will go there to hide, from other predators, because fish that use gills to breathe in the ocean need the oxygen from the water. A seal doesn't, carries the oxygen with it. So we think that what is happening is that these are areas where the animals are going after fish that have been slowed down because they're avoiding fish predators in the oxygen minimum zone, but it makes them very good prey for things like elephant seals. So that was an elephant seal that just using this little camera ate fish. Here's one where it's a squid. And I admit it's hard to tell exactly what it was, but if you do it in slow motion, you can see that it was squid. And then here is, we don't know what. And here's a shrimp. These are pictures taken hundreds of miles at sea in 400 to 600 meters of water. So just, that's a pretty, you think, oh, cool picture, but then you realize where that picture was actually, that video was actually taken. And here's the jellyfish. I don't think the animal was eating it, it just came past. So that gives us some ideas about what's going on. And one of the things that's an advantage of having these large data sets is you can start to see when things become different. And one of the things that we started to notice, well, sort of set the stage by talking about work that's done at Mbari, and they've been following all kinds of things that are happening in Monterey Canyon, Monterey Bay for quite a while. This is a study of Humboldt squid. These are those very large squid that live predominantly off Baja California, and occasionally they come north. And so here is, they have hours and hours of video with their deep rovers, their deep ROVs, and you can see there's no Humboldt squid over this whole period from the 1980s to the early 90s. 1998 was an El Nino event, and that was the first time we saw Humboldt squid in Monterey Bay. And then 2005, we got much more Humboldt squid in Monterey Bay. So there was this increase in the abundance of Humboldt squid. We had this one female that had this very unusual pattern. Remember, all the images I showed you so far showed the females out here. Well, here's this female that stayed right inshore, and she spent most of her time feeding right here along Monterey Canyon. And this is that record again. Spent most of her time here, never left the California current. This was incredibly unusual. At the time we did this, we had 300 tracks. We now have 600 tracks. This is the only female that's ever done that. When my students told me about this record, I said, oh, it's a young female. She doesn't know what she's doing. Well, she was a 7-year-old, which is prime age female. Then I said, okay, well, she's going to have really bad foraging success. Well, she had one of the best body mass changes, best mass gains of any animal we'd measured. So obviously she knew something, what a good deal was. Well, if you look at her dive pattern, which is, here's her diving pattern because we recovered the instrument from her, and we plotted the diving depths of that female. And this is from the Mbari study where the Humboldt squid were located. You see pretty amazing correlation between what that female was doing and what the Humboldt squid were doing in Monterey Bay. So what's interesting is Humboldt squid are coming up from Baja California, and we started putting satellite tracking devices on females down in off Baja California. And what we found is that many of the individuals come up and feed where our elephant seals from Año Nuevo are feeding, but about half of them stay there. And I'm starting to speculate. I don't know what these females are doing, but I wouldn't be surprised if they're feeding on Humboldt squid. This region of Baja California, I found out from colleagues in Mexico, is one of the prime areas for the breeding grounds of Humboldt squid, and Humboldt squid are very common here. So I suspect that what we see here that's very different than what we see off California, because the Mexican elephant seals are taking advantage of these large aggregations of Humboldt squid. They're capable of doing it when we see Humboldt squid actually coming into Monterey Bay. Now, again, I mentioned, we have a lot of these records. And so one of the things we've done is we both on purpose, but also started to realize that we've got females that we've looked over multiple seasons. And so here is a female over 3 successive years that we're able to follow her tracks to see what she was doing. And so you get some females that have incredible site fidelity. This is a female that was much more variable, and this is a female that did something different than the one down below, but also was similarly quite similar. So this is 1 year after another, looking at the same foraging trip. This is our amazing record. My students were looking for 7-year-old females, and they, wrong way, and they accidentally put a tag on a female that, they read the tag wrong. They put a tag on a 17-year-old female, which is pretty old for an elephant seal. We normally wouldn't tag a 17-year-old. But what's amazing is by accident, they picked a female and put a tag on her, who we had also put a tag out when she was a 6-year-old in 1995. So this is a female, the same individual as a 6-year-old in 1995, and by accident, we selected her for tagging 11 years later and put a tag out. And lo and behold, she was doing the same thing 11 years later. Now, we don't know what she did in between, but I suspect that she probably was doing the same sort of foraging pattern. So then the question comes, well, why do you have, what's the difference? Why are some females doing this high site fidelity and others low site fidelity? And to try and explain this figure, these are females that are doing this high site fidelity, doing the same thing year after year. And this is how variable, how stable, or how variable the ocean conditions are. So this is chlorophyll content in the ocean. This is sea surface temperature. So out here, it's very variable. The females who have low site fidelity are feeding in areas of the ocean that are quite variable. These females that are feeding in the same place year after year are feeding in regions of the ocean which are quite stable. And so this is sort of reassuring in one respect because things are changing, the climate is changing, and so the advantage of doing the same thing year after year is where oceanographic conditions, the climate is stable. The advantage of doing something in a variable and not being sort of stuck in one pattern is that if things are changing, you're going to be able to change with it. And so what's reassuring is that not every female out there is stuck doing the same foraging pattern. There's a group of females which are able to respond to these quite variable situations. Now, the other aspect of this, which one of my graduate students, Rachel Holzer, is interested in is there a personality trait? Are these females that do the same thing year after year, are they risk averse? And are these females risk tolerant? And so she's looking on some behavioral methods to study whether there is a consistent personality, quote, associated with these different foraging patterns. Okay, so that gives you sort of this broad overview of what we've learned and how we got there. I'm going to spend a little bit of time telling you about how we can now use these animals to study the environment that they live in, studying the ocean. And this is, again, a female where it's a repeat female. Remember I talked about we had an El Nino year. We had that 2005, which was a warm water year. Some of you may remember a few years back we had the hot blob in the ocean. Well, this was that warm region of the North Pacific, and our elephant seals were swimming right through that. And so our elephant seals are really good oceanographers in that they're measuring the ocean environment in this region, which a lot of the equipment that we use, Argo buoys and the like, completely missed that. The Argo buoys were out here on the outskirts. So we're able to look at what this female did from one year from the next. We could see how whether she was gaining more or less mass, whether she pupped or not. This was a different female that, again, you can see looking at 1 year from the next. And some of these female, and you can see how their foraging pattern changed as a function of this changing climate. So we're able to see how these animals respond to these different climatic events. And what we found is that some females do okay and others are more sensitive. I'll get back to that in a minute. But here's all those tracks. Here's that warm blob. And we have many females that are swimming through this area. And every one of these females has a temperature profile like this, where this is her surface track, And then she's diving down, and these females have temperature sensors on them. And so this is the temperature profile of the ocean monitored by an elephant seal. And you can see that warm blob here. You can see the warm blob actually descends down and through the water column, and sort of that warming associated with that oceanographic feature. This shows the diving pattern of that animal. And so this is the nighttime dives, and this is the daytime dives. And you can see that there's a slight deepening of where the animal was feeding in that warm blob region. And so this is work that Rachel Holzer is following up to both use these data to study what the elephant seals were doing, but also to study the oceanography and figure out the heat content and how much heat that was actually in the ocean associated with that warm blob. Now, one of the interesting aspects of this was when you've been studying a population for so many years, you have a lot of data on what we've looked at females and the size of the pups over one year to the next. And so typically you see females are giving pups that, or these are females that, how they come back at the end of a trip. And all of a sudden, this is the warm blob. You say, oh my gosh, look, the females must have been doing better during the warm blob, but that doesn't make any sense. And what we realize is during this time when food was not available, the smaller females didn't even bother. So the smaller females didn't even come back to the beach. And so unless we had the context to know how many females were coming back and who did come back and who didn't, you might have the wrong impression that things were actually the opposite. And what this means is that big, healthy, experienced females are able to deal with these oceanographic events. The younger, smaller females, the inexperienced females are not, and they don't even bother to come ashore to breed.
So does that mean that they don't come back at all, or that they perish?
They probably will come back another year. So if you look at the other, I already talked about this site fidelity, and so you might ask me, well, how does this figured out? And so again, because we've been studying these animals for so long, and for we've started to get generations of individuals. This is the track of a mother in the sort of purple, and this is the track of her daughter as an adult female. And so this is another individual of the mom's track and the individual of her daughter's track when that daughter was an adult. So we now have about 40 or 50 of these tracks that we're just starting to sort of piece this together, and it allows us to ask the question as whether these animals have a genetic component to their foraging pattern. We've also had where we've tagged females as a weanling. Here's a weanling animal tagged in 2008, and this is that same individual 4 years later as an adult. And so we've been increasing to ask the question about what do these animals do? How do they learn how to forage? How do they get the programming? Is it genetic? Is it learned? And trying to tease that apart. And I don't have an answer to that yet. It's just sort of giving you a teaser for what where we're going with this research. Okay, so I told you 3 unique places. I started in the Antarctic, took you to Año Nuevo, which is a place that's been very special to me for basically my entire career. And now I'm going to take you to the Galapagos, which is a place that I actually went to for the first time with Bernie LeBoeuf when we were going to study. We had the good fortune of having a boat that we literally surveyed every island except the southern island here. So when I say I've been to the Galapagos, I've seen almost every island from a small boat over a period of a month, staring at it through binoculars. But for me, the really interesting reasons to go there is the Galapagos sea lion and the Galapagos fur seal, which I'm going to give you a little bit of a discussion of work we've done. We were interested in using some of the first time depth recorders, satellite tags that had a GPS tracking component to them. And here's the traditional technology of a satellite, Argo satellite location of a Galapagos sea lion. And now here's that same individual, which is now tracked with GPS. So this is a device on the animal, and I'm comparing, the white is what the Argos positions tell us, and this shows us the actual positions because the animal had a much more accurate device in the form of a GPS tag. And so one of the reasons we go into Galapagos is this picture of that sea lion. I'm probably standing at least as close as I am to any of you. These animals are very easy to come up to approach. They don't run away. You can put tags on them and you can actually watch and see what they're doing. So some of what we've been able to find out is that these are animals, different individuals that were tagged right here. This is Santa Cruz Island. If you fly into the Galapagos, you fly into this island, which is Baltra. The green are shallow diving individuals. The red are individuals which are diving to the bottom, and the yellow are animals diving into the deep water column. You can see the different dive patterns here. These are the shallow diving individuals that only dive at night. These are individuals which are diving during the daytime but diving deep into the water column. And these are the individuals which are diving to the bottom and feeding on different kinds of fish on the bottom. So this is the kind of work we can do now that we can put these tags on sea lions. And we can, looking at the Galapagos fur seal, which you see here, Galapagos fur seal mother and pup, Galapagos sea lion mother and pup. And now we ask the question, okay, we've got these two different species. How are they partitioning the habitat? And the blue is what the fur seals are doing. The red is what the sea lions are doing. And this shows the blue tracks of fur seals, the red tracks of the sea lions. And it's starting to fit this pattern because we've studied sea lions in one place, we've studied fur seals in another, but this was the first time we were able to put the two species together to see how they're partitioning the habitat. And what we find out is that the sea lions like to feed on the continental shelf and that the fur seals like to feed offshore in the more open ocean, shallow or surface layers of the ocean. And so this is sort of an insight into how they partition that environment. Now, the one thing that we started to realize is let's compare the sea lions in the Galapagos over here with all the other sea lions in the world. And so we took what we learned studying elephant seals and the technology developed with elephant seals, and through our own research and then coupling that with a few other studies, looked at the sea lions throughout the world. There's only 6 species of sea lions, but still they're scattered around the planet, and asked the question of, are there differences in how the way that they spend energy? And the Galapagos is a very warm place. We studied first the Galapagos fur seal, then the Galapagos sea lion, and all I really want you to take home from this figure is that the average metabolism, the sort of intensity of life, is much lower for the Galapagos fur seal and sea lion than sea lions and fur seals anywhere else on the planet. And that fits. We all sort of like to take it easy when it's really warm, and that seems to be the case for these guys. We then looked at the diving behavior. So here's how long the individual different species dive, and this is how long they dive, and this is how deep they dive. Now what's interesting is these are the big sea lions. This is the Steller sea lion and the southern sea lion. It's the Australian sea lion, and here's our little Galapagos sea lion. The Galapagos sea lion is the smallest sea lion. What I haven't explained to you yet is the bigger you are, the longer you should be able to dive. And so these guys should be the longest, deepest diving sea lions, but the big boys are the shallowest, shortest diving sea lions. This little Galapagos sea lion is really a champion diver, and it's diving longer than any other sea lion, but it's almost diving as deep as the New Zealand sea lion. And that sort of didn't make a lot of sense to us in terms of how does it do that? Sea lions have a certain amount of the internal scuba tank that a sea lion has. It has a lot of muscle, it has myoglobin in it, they have larger blood volumes. And when we plotted out how the dive duration and the total oxygen stores, so what we've measured here is how much muscle oxygen is stored and how much blood, that little Galapagos sea lion is a champion among sea lions. In fact, if you'd given me these data, I'd say we're looking at a seal, not a sea lion. So this gets to the question of how do these different individuals make a living? What do they do, and what is the physiology that allows them to do that? So I'll end by just sort of giving a plug to one of the things that we're trying a new thing at UC Santa Cruz, and that's to pair people up with some of us researchers in places that we've worked. So this coming October, we're going to do a trip to the Galapagos, and these are all pictures that I've taken on a previous We'll first take a little spin to the Andes, to Otavalo, which is a very interesting market for the local Andean people. Then fly to the Galapagos, where of course you'll see the Galapagos tortoise. This is Bartolome. We'll see that. It's a really interesting, beautiful place. Spending time on a boat, checking out the sights. This is Daphne Island, where a lot of amazing work on Galapagos finches has been done. And then see some of the critters, the iconic Galapagos marine iguana, and just amazing seabirds in general. And one of my favorites, the Galapagos penguin, which is this guy here, and masked boobies. And of course, and then the flightless cormorant. You can see that there's got little tiny wings, and of course, my good friend, the Galapagos sea lion. And there's lots of frigate birds. And I'll leave it at that. Thank a lot of the people who've made this funding available, and we'll see if there's any questions. And I'm sure Carolyn will also answer any questions. Yeah.
So, could you talk a little bit about the tracking devices and how they're attached and all the different components that are in there, all the different things that you're measuring with them?
So we attach them with 5-minute epoxy. It's just you go to your hardware store and buy 5-minute epoxy. When we first started doing this, we didn't know what we would put harnesses, or we come up with all kinds of other things. And one of the things that's made my life easier studying seals and sea lions is they have hair. And you can glue it to the hair. So you're gluing it to the hair, and when the animals molt, it falls off. So if we don't recover the tag, it will fall off. Cetaceans, you can ask Ari Friedlander about how they do it with cetaceans. It's much harder to get tags on cetaceans. So we glue it onto their hair. The simplest tag measures time and depth. So it has a pressure transducer that just measures depth at some frequency. The next thing we added was temperature. We added light level. And now we have tags that measure salinity temperature as a function of depth. We have a tag that has a fluorometer built in, so we can look at chlorophyll content of the water. I have a graduate student that's working on putting an oxygen sensor onto the tag so we can, I told you a little bit about that O2 minimum zone. We want to measure and see if the animals really are feeding in that O2 minimum zone, and to correlate the actual O2 content of the ocean with the foraging behavior. We have tags that have 3-axis accelerometers. I mean, our cell phones have a lot of technology that we're sort of taking because when it's mass-produced, it's cheap, and we incorporate that into these tags. Early on, I showed you some tags that have O2 sensors. We have tags, blood oxygen levels. We've got tags that measure heart rate that are routinely being deployed. There's all kinds of, there's camera tags. So, we started very simple, and it's really a matter of taking, it's very difficult for us to build new technologies, but we can take technology that's already off the shelf and that's inexpensive and apply it to our animals.
So the life of each device is lifetime of each device?
What is the lifetime? Battery is probably the biggest problem is the more stuff you put on it, the more power it consumes. And so most of the tags will last, a year is the longest anything will operate because it'll fall off the animal. So if it lasts a year for the studies we do, that's as good as it needs to be.
I have a question for Ms. Casey. In the slides that you showed us where you used the vocalizations with the speaker box and the elephant seals responded.
Yes.
I found it really, what struck me about that was that, aren't they using their eyes? Can't they see it's a speaker and not an elephant seal? And so that means to me that, gosh, they are almost totally dependent on this vocalization to understand this threat, this retreat or approach thing. Can you comment on that, please?
Yeah, absolutely. I'll just repeat the question for the live stream. So could you hear it? Okay, cool. Yeah, it's a really good question. And so remember that the signal is multimodal, meaning that there's an acoustic component, but there is a visual component. They're sitting up, and there's this very stylized posture where the males are sort of showing their profile. And so we haven't asked the question of whether they can visually discriminate between individuals. I really want to design a study where we have like a pull-up of a male that we like display to another male. So it really was a question of, can the call itself represent the individual in the absence of any other, visual component of the male? And the answer is yes, but I do think that they're dependent on both. I wouldn't say that they're exclusively dependent on the acoustic component. That's just what we tested, but it's enough to elicit that response. And when you think about when these animals are most active, because it's really hot during the day, they tend to kind of sack out. And then in the morning, when it's low light conditions, and in the evening is when they're most active. And so they can really move each other across big distances where they might not be able to detect visual differences between individuals at those times of day, but acoustically they can do it. So I think they are using both, but definitely the acoustic signal is really important.
Do you find any evidence of acoustic mimicry, where betas are mimicking alphas?
You want to join our research team? And that's like my life's favorite question, this idea of some animals sound like an animal regardless of where they're raised. So if you take a dog and raise it by itself without exposure to other dogs, it's still going to sound like a dog. Humans, dolphins, elephants, and some species of birds are what we call vocal learners, meaning that they have to be exposed to other conspecifics in order to learn how to sound like that animal. And there's some people who previously thought that that was the precondition for human language. So it was this very hotly debated topic. And then we started to show that different animals were capable of doing that. In order to answer that question, you need to do one or two things. You need to, one, track individual animals in the field over the course of their vocal development and know what the acoustic environment is for that individual to see whether or not they're emulating certain signals. And so we're in the process right now of tagging little guys, tracking them over their lifetime, and then also kind of monitoring who they're interacting with to see if they do sound more like the most dominant individuals within their social network. Or alternatively, you could take an animal, raise it in captivity, and see if it still sounds like an elephant seal despite never being exposed to other individuals, or alternatively, produce an aberrant call. And we did have one female northern elephant seal at the Long Marine Lab who was raised without any access to other individuals, and she sounded weird. She had like a Tarzan vocalization that she made that I'm sure Dan remembers. And so I personally think, without any scientific data, that they do learn their signals, but I'm trying to kind of investigate that, and it'll probably take a long time.
I have a question for Dan. Something you hinted at, but I was wondering if you could amplify on, is the use of marine mammals, whether as volunteers or conscripted, to do oceanographic research or provide data not for use by marine mammal scientists, but oceanographers.
Yeah, it's a very active area of research. And so the first thing I'd say is that we're underfunded in terms of our ability to track these animals. And so any help we can get putting tags out on animals and seeing where they go is great. The reason I say that is that we have now we've collaborated with oceanographers, physical oceanographers, and they get excited about they now call us up and say, can you come and put your tags out here or here? And in the Antarctic and in the Arctic, that is I would go to a meeting of glaciologists, people looking at the ice fronts and glacial melt and whether the ocean is eroding. And they want me to come and put my tags out on animals. And in fact, there are colleagues that actively take a number of our colleagues. And so a lot of our research in the Southern Ocean now is funded in part by collaborating with physical and biological oceanographers. So it's a two-way street. We collaborating with a physical oceanographer, we mapped the continental shelf of the Antarctic Peninsula using our elephant seals dive to the bottom, and so we were able to use them as depth sounders. And so wherever they dove, they often dove deeper than the known bottom because it wasn't very well surveyed. And so we knew they weren't mole seals. And so we were able to find where there were troughs in the ocean, which actually turned out to be where warmer, deep water was coming up and eroding the ice front that was degrading faster than the atmospheric climate heat budget would have told us. So to me, that's one of the exciting things. And as I said, I'll go to a meeting in the Antarctic, in the Antarctic community, and there'll be people presenting physical oceanographic results that have been collected from our seals, and the same for the Arctic. And the reason that's so important for them is we don't have the technology to go into the pack ice and routinely sample other than with a ship. And ships are incredibly expensive, and seals are cheap to, they feed themselves and they carry the tags almost for free.
We have 2 online questions. First one's from Tom. Have you done any studies when the elephant seals are hunting out in the ocean around vocalization? And is it social and collaborative or competitive?
I guess that's a question for both of us. I mean, we've recorded underwater hydrophones around the colony, which was a scary endeavor because we were wading out into the ocean and dropping recorders as animals were coming ashore. And we did find that when they're close to shore, they are producing calls underwater, probably practicing, and sometimes they fall asleep and they blow bubbles and they vocalize. But once they leave the shore, they're fairly solitary, and we think are hunting solitarily.
Yeah, and we were quite interested in that question some years ago, and that was my first foray into bioacoustics. Back then, they didn't have really nice small acoustic recorders. We took a Sony digital audio tape recorder and put it in an underwater housing, and then put it on the back of a seal. And then as technology developed, they got smaller and smaller, more sophisticated acoustic recorders that we deployed on these animals. It became very boring because we got no vocalization. We had nothing from an elephant seal. So what was interesting, though, is we did get, from an elephant seal recording, we got humpback whales, and then one of the other things was interesting, it was a sperm whale. So some colleagues have said, elephant seals may not make any sounds, but wonder if they passively pick up on, if there's a sperm whale, over there, will an elephant seal go to where that sound is and maybe figure out that there's a good thing to eat over there? We don't know, but the answer to the question is they don't vocalize at sea. They could passively use sound, though.
And our second question is, what do we know about how seals find their way back to the rookery year after year?
Patrick Robinson, who's our reserve manager, did his doctoral work on navigation, and we know that these animals follow great circle routes, which means they have to have a map sense of where they are. They're not just using a magnetic orientation. But we also know that they follow the Earth's magnetic field because you can look at the tracks relative to the Earth magnetic field. So we don't know exactly how they do it, but everything that, when you analyze the pattern of the tracks and the movements, it's very consistent with using magnetic fields. There's also the possibility that they use visual cues like position of the sun or stars. We know that birds do that quite well. So what we have is the descriptions that say they're doing something really interesting because well, you could see by the tracks I showed, those animals know they're going back to Año Nuevo. So we know they do it. We know they do it consistent with magnetic fields, but that's about as far as we've been able to take it.
I was curious about the process dives. Are they done pretty much at the same time of day or not?
That's a really good question, and I honestly don't know the answer to that. I'll have to look at that. We've always looked at that they come in, when you have lots of feeding dives, they're followed by a series of processing dives. So my guess is that they're independent of day length, but I don't know that we've ever actually looked at that, and that's a really good question. I'll have to go back and check that out.
Do you think the seals and sea lions are would be affected by underwater sonar testing like the Navy wants to do? And how do you think that might affect them as it affects like marine mammals who use echolocation?
Yeah, I think that's a question that both Dan and I are working on. My postdoc now is looking at the effects of sonar on dolphins and other small species of cetaceans around the Channel Islands. And I think collectively, animals being exposed to sounds continuously, it has the potential to have really negative impacts, either to deter the animals from feeding in a certain area, maybe it surprises them. And so there's a ton of research going on in this field right now to that respect. And so as far as seals go, we wanted to study that in seals. It hasn't, as far as I know, been included in with respect to military sonar, but I do think that it has the potential for, after continuous exposure, to probably prevent animals maybe from coming back to a similar area, right? Because it's either annoying or startling. But Dan probably has some thoughts on that as well.
Yeah, in most cases, the sound is not going to be immediately detrimental. You have to get really, really close for it to damage their hearing. But there is behavioral changes. And as Carolyn said, they might leave the area, they might stop feeding. And so a lot of the work we're doing is trying to figure out how much of a disturbance in terms of whether it puts them off from feeding or it moves them to a poor foraging area is too much. And so the immediate answer to the question is that it's not devastating to these animals, but it's like, is it death by 1,000 cuts? And so that's really where a lot of the research is, because, for example, in Southern California, there's been a Navy sonar range there that they've been testing ships and sonar and do anti-submarine warfare training for years, and there's still a lot of marine mammals out there. But we don't know if they've somehow acclimated to it, whether there's fewer individuals there, and it's hard to study these animals because they're way offshore, and Ari Friedlander is amongst another group of people that are studying these animals in that region. Carolyn pointed out that we can't seem to get anybody that concerned about sea lions and seals.
They're the
much more
second-class citizens of the marine mammal world.
We've been trying to get people to be interested in, well, what about sea lions and what about elephant seals? They tend to be interested in the dolphins and the whales.
You have a question on whether or not there's been enough, you have enough resolution, enough time has passed to be able to see any time dependence in the migration patterns, either due to sort of natural cycles in oceanographic characteristics or, climate change.
No. Well, yeah, yes and no. We've studied animals over acute windows of time, like the 1998 El Nino, where it was a very dramatically bad year, and that's kind of a climate signal that's very short term. We've been following the pupping rates and the massive pups from the early 70s to now, and we do see that the massive pups is a little bit lower, but that could also be because the population is recovering. And so as you reach carrying capacity, you're going to naturally reach, there's less food to go around for everybody. So what I can say about elephant seals is that they've already gone through one of the most amazing bottlenecks, going from less than 25 to 50 individuals in 1900 to over 250,000 today. Will climate change affect them? Probably. Will it be debilitating? No. They're probably so resilient, and the way everything they do is so robust, and they can use so many different parts of the ocean, that they're an animal that will probably be fine. They may have a lower population, they shift their populations, but they're a really robust animal. So yeah, I guess I'm not as worried about elephant seals as I am about some other things.
So I have a question for Caroline.
Yeah.
Do you have a hypothesis about why the different populations do you have a hypothesis about why the different populations in the different locations in the 60s all sounded, they had very specific dialects that didn't vary among those specific populations?
Yeah, it's a good question that we still don't have a lot of resolution on because we weren't there. But if the animals are learning their sounds, then you can imagine a very small, so say maybe there's a few individuals that recolonize a site. So it's me and Dan who show up, we're males, and then there's one pup that's born horn there. He's probably either going to sound like me or Dan, and collectively we're going to sound all pretty similar to one another. And so I think that this was the case at most of the sites, that just by chance, maybe animals at different sites, the original founding fathers of those elephant seals, sounded different from one another across sites. And then as animals grew up and generations kind of grew, animals tended to entrain to the signals that they were exposed to at those particular sites. And then over time, if you are imitating someone, you might imitate them but add a little mistake, or what we call like a copying error. So I want to sound like Dan, but then I sort of add this hiccup at the end, and then that's my call. And so it took probably several years for those mistakes to kind of proliferate into the colony, and then those mistakes actually served a purpose because it helped individuals sound more recognizable. So I think it was just the founder effect that animals happen to sound more similar to one another within the colony and different between, and then that was actually not functional for them in the long term, and so it changed over time. But that's completely, I don't know the answer. That's just my guess.
So I have another answer to the climate change thing. Specifically elephant seals, there's not an issue. But if you go to the East Coast where northern right whales are, the population is changing and they're shifting more to the north as the climate is changing. And what's interesting and problematic is that they've got all kinds of fishery that right whales get caught in fishing gear. And so they figured out how to deal with that in the Gulf of Maine, and there's a lot of good protection. The right whales are now moving north, and there's plenty of food. They're following the food, but now the fishing regulations are not in place to protect them. And so we're now starting to get, once again, a lot of right whales getting entangled in fishing gear. So that this climate change thing is insidious in that you can have a shift, and they might be able to accommodate that, but then it may put them in different jeopardy from interfacing with humans. Also, there's people, again, with bioacoustics, have been listening to the sounds of whales in the Arctic for many years, and they're now seeing humpback whales and fin and blue whales higher and much more into the Arctic Ocean than we've ever heard before. So there are these changes. We've also seen a gray whale off the coast of South Africa and a gray whale off the coast of Israel. The gray whales were exterminated in the Atlantic 300 years ago, 200 years ago. So there's no question that those gray whales had to come over through the Arctic Ocean. So with elephant seals, we haven't seen anything, but in marine mammals as a group, we're starting to see changes in what they're doing in the environment. I had to put my other hat on. Yeah.
I don't know if this is a question or comment, but going back to the question about whether climate change is the effect that we've said that elephant seals are real robust, it seems like there could be another factor that could be negative there. Is you've gone through this tremendous bottleneck of decreased genetic diversity. And if genetic diversity allows this population to expand into different climates, it seems like that could be a very negative effect.
Yeah, the question, I'll just restate it, is about what's the effect of the genetic bottleneck, and how does that, we've all worried about that in terms of elephant seals are probably, well, people have done genetic studies on them because they wanted, for the kind of work that Carolyn was doing, wanted to easily determine paternity. And so whereas any other mammal, you can go out and take a bunch of males and just grab some DNA and then just blindly pick the father. With elephant seals, they're all brothers and cousins and nephews, and I mean, because they're probably, it could have been one male that was the father of all current northern elephant seals. So they're incredibly inbred. They do have, we do know the more sophisticated genetic testing, we can start to tease apart individuals, but they're all very, very closely related. That makes us concerned, as you pointed out, that, gee, they may not have the genetic variants to respond to these changing habitats, to diseases. That's been a concern that we've all thought about when the first paper came out 30 years ago that showed these guys were all very closely related genetically. That's a concern, but so far everything we've seen about this animal, there's nothing that shows us that it's a concern, but it's not been a realization. So the good news is right now, seems that whatever that bottleneck is, it hasn't hurt them. But that's one of those caveats, it's like right now.